Hey everyone, I hope you guys are having a good time at home or school or wherever you are. Hope you guys are staying happy and healthy, but also up to date on all of your schoolwork. So last week you guys had an exam, and I hope you kind of are able to see where you're at with this online course. I hope that if you need help with the transition or any of the information, please don't hesitate to reach out to me. And this is just a continuation of the previous video, and I may post one next week if I have time. But really, again, these are for reminding you of things that are due, what's coming up, and about SI sessions. So the 2 sessions are going to be Tuesdays and Thursdays still at 2 p.m., and there's a Zoom link that I sent out with that information. So if you have trouble getting on or can't make the session, just email me and let me know, and we can work something out. The next thing I'd like to point out to you guys is your schedule for the rest of the semester. So you guys are going to be covering things like the cell cycle, mitosis, meiosis, and cell communication. And Dr. Temple has already posted videos online, and basically he'll go through the PowerPoint, write notes, and explain what's going on. So if you have any trouble with the information, please take a look at those because they're very helpful. Also, you have a final exam Tuesday, April 21st at 8 a.m. Keep in mind the final SI session is on April 14th, so if you need help between now and then, please try to make the SI sessions whenever you can. So the first thing I'd like to talk about is the cell cycle. So we have different phases. You'll have to know generally what happens in each phase. So G1 is more like growth and development of the cell. The S phase is where DNA replication occurs. The G2 phase is another growth phase. Those three comprise of what's called interphase. And then the M phase is where you have mitosis and cytokinesis. So key difference here, mitosis is nuclear division. So you're starting with one nucleus and splitting it into two. And cytokinesis is when you're splitting the cytoplasm. You'll also see Dr. Temple talk about the transitions between each phase. So they're basically checkpoints that certain things have to be done correctly before the cell can move on to the next phase. So those are important to understand as well. Next, I would like to talk about the amplifying caspase cascade. So caspase is a molecule that activates apoptosis or cell death. So it's stored in an inactive form and becomes activated. Each type of caspase that's activated will cleave a certain structure or protein. So the more activation you have down the line, it'll basically start degrading all of the components of the cell, and those components will get reabsorbed into the bloodstream. As far as I remember, Dr. Temple won't ask you the name and detail of everything on this slide. It's just important to understand that caspase is stored inactively. It becomes activated. As one is activated, it will activate many more, and eventually the cell will be degraded. Another important thing to talk about are mitogens. So these are molecules that, as you can see in the image, are going to attach to mitogen receptors, and basically this will activate a signaling pathway. This will start or initiate the G1 and G1S CDK. So essentially what that means is they're going to start certain phases of the cell cycle, and then other proteins will also get activated through phosphorylation, and those proteins will lead to transcription, translation, and cell proliferation. So basically, all this slide is showing you is that you have a molecule that's initiating a signaling pathway that will activate the CDK to activate other proteins and start the process of making proteins and other structures that the cell needs in order to consistently divide. So the next thing I'd like to talk about is what happens when DNA is damaged. So the first step shown is when a protein called P53 is phosphorylated, so it becomes stable and active. And once that protein is active, it will initiate the transcription of the P21 gene. So then this P21 gene's transcription will eventually lead to the creation of a P21 protein, and that protein is a CDK inhibitor. What that means is once that protein is made, it's going to make any CDK molecules inactive. So if those CDK molecules are inactive, they can't continue the cell cycle. They won't be able to initiate that next phase, whether it's G1 or S. Alright, so that's the end of the video. If you guys have any questions, comments, or concerns, please email me. I hope you guys are able to make use of the resources that Dr. Temple is providing you. Have a great week.